Our immune system guards the body by recognizing and defending against bacteria, viruses, and other harmful invaders. But the immune system can overreact or start attacking the body's own tissues, leading to diseases. For the past 10 years, Professor Feng Shao's team has been focusing on unraveling the molecular mechanisms underlying host cell inflammatory responses.
I was studying how does a particular bacteria cause disease or infect our body or our cell. And we know that different bacterial pathogens, they have different mechanisms. So what we learn from there cannot be generalized to the broader level. As a scientist, we always want to look for something new and do something that we have never done that will get you more excited. So that's the reason I decided to shift to the innate immune research. This system works broadly to detect all kinds of bacterial infection and even other microbial infections, so the results could have a bigger impact. So LPS, which we call lipopolysaccharide, is a large molecule, is the most abundant structural component of the cell wall of gram-negative bacteria. In our field, LPS is considered the most important molecule for bacteria to survive, as well as a major molecule detected by the immune system. Because as the immune system, we need to detect the infection. And the way to detect the infection is to recognize something abundantly.
How LPS is recognized in host cells was not clear, and the responsible pattern recognition receptor was unknown. Shao found that inflammatory caspases, a family of proteolytic enzymes that can induce cell death and inflammation, caspase 4 and 5, are intracellular receptors for LPS. They can directly recognize LPS and become activated, then triggering host immune reactions. He also discovered that gastamine D is the downstream death effector of the inflammatory caspases.
There is a family or a group of proteins that all share the pore-forming activity. We call it gastamine family. When they are activated, and they will also do the same thing to make pores and to kill cells by pyroptosis, which is the pro-inflammatory form of cell death. Conceptually, the finding actually is paradigm shifting because previously on studies on cell death are focused on the caspase. So the discovery of gastamine D means that there are molecules downstream of caspase. Those molecules are executor proteins in the cell death pathway. So that changes our mindset about understanding the execution of cell death.
By manipulating gastamine proteins, it is possible to control inflammatory responses of the body, paving the way for developing potential drugs for serious inflammatory diseases and possibly cancers.
The discovery of gastamine essentially provides a novel drug target to manipulate the inflammation. So if we block gastamine activation, that might help provide some clinical benefits in treating inflammatory disease like sepsis.
Shao tends to question the conventional beliefs in biology. This mantra helps him find the necessary breakthroughs in identifying caspases as the LPS receptor. With a team that has diverse backgrounds, Shao's team is unafraid to try new experimental methods and tools. For instance, the discovery of gastamine protein involved the use of CRISPR-Cas9 genome editing technology, which was quite new when first applied.
So I came from a chemistry background and gradually moved to biochemistry, biology, and now immunology and medical science. The direction of my career pursuit has always been to do something useful in clinics to help us to understand disease and treat disease.
The 2019 Future Science Prize in Life Science was awarded to Feng Shao for his seminal discoveries of cytosolic LPS receptors and downstream effectors in inflammatory responses to pathogenic bacteria.